Welcome back to another session of RNA biology, and we were discussing about the advantages of having intron, and the answer is introns are indeed important in multiple ways. One is for expression of genes and also for creating diversity, plenty more, and we will see them, address them one by one, because in a nutshell we can say introns are extremely important, and eukaryotes, credit goes to introns to a great extent compared to prokaryotes. So the biological advantage of alternative splicing is that two or more polypeptides can be derived from a single gene, and two or more is a very small or silly number when you are going to see a real time example. It will baffle you completely. But even 2 is definitely more than 1, right? So 2 is definitely handy. So alternative splicing minimum assures that there are 2 proteins coming from 1 gene, and hence with less number of genes you are able to do more. Coming back to the example which I told you, with assumed 23,000 genes you are able to perform the task of, or you are able to make a complex organism that will require at least 1 lakh genes. Is credit goes to alternative splicing, and it is an enigmatic phenomenon. Nobody completely understands the real essence of alternative splicing, and I am sure in future someone may be able to come up with a clear-cut answer to alternative splicing, and that will be one of the Nobel Prize winning discovery, because alternative splicing is that important, and we do not even now completely understand how many expressed genes are there in humans. We don't know. All we know is the possible coding sequences which can act as gene from the entire genome. So total number of genes expressed in the lifetime, because some genes are important only during developmental stage. After that they are permanently shut off, they will never turn on back again. So it is very tricky and very difficult to identify that which are the genes, what are the possible alteration or alternative versions possible from a gene in a simplistic form, especially in human. This allows an organism to carry fewer genes in their genome, because during evolution has happened in both directions. In some direction the genome size started increasing, in another direction the genome size started decreasing. Genome means you may wonder what is this genome you are talking about. Human genome is around 3000 megabase, means one base. Once it becomes 1000 base, it is 1 kilobase. 1000 kilobase is 1 megabase, and we have 3000 megabase is our genome, whereas E. coli bacteria has got only 4 MB, 4 megabase. Drosophila and zebrafish, all these animals have got differing size of their genome. Like some animals have got extremely large, say for example axolotl, which is a model organism which has got 12 times bigger than human genome. That means 3000 into 12, that is the size of axolotl genome, whereas zebrafish have got only 1700 MB, which is almost half of human genome. But remember, they all have got more or less the same number of genes. Don't think fish have got less number of genes than human because it is very small, not like that. Size, or elephant have got larger number of genes, or dinosaurs have much bigger number of genes than a wall lizard. So these are all completely different events which you should understand. The size of an organism is not. Some worms have extremely large genome and they have large number of chromosomes also. So evolution has happened in both directions. So we cannot simply say in one answer, we cannot say more genome is more complex organism, more genome means more genes, More genes means you can have complex or more ability to perform larger number of functions. Not like that. It always depends which organism you are talking about, which gene you are talking about. And also you should not think every gene have got at least 5 alternative spliced form. No, there are plenty of genes which have only one alternative splice form. And we should not think every human gene have got introns. There are many intron-less genes also, somewhat like bacteria. Just one exon, that is the gene. So this is also possible. So there are less number of concrete rules especially in genomic research. Let us see one example how this alternative splicing is bringing in complexity. Sex in Drosophila is largely determined by alternative splicing. Let us quickly see an example. This gene, SXL gene, which has got exon 1, exon 2, exon 3. And if the splicing happens between exon 1 and exon 3, then you end up getting a female. If splicing happens between exon 1, exon 2 and exon 3, you end up getting a male. So this is one easier example to understand, because many organisms do not have a dedicated sex determination gene. Even if it don't have, it will be able to perform a differential function during the development and subsequent animal physiology. It will be able to bring in like human body by default is female in structure. So the role of the testosterone or the hormones is to resist or fighting 24/7 not to become female body. So if a organism, human, is born and that person don't have hormones, testosterone or male hormones are not there, or male hormone have got its corresponding receptor, if that receptor is mutated, you cannot distinguish that person from a typical female. No way you can distinguish unless you do anatomical study or chromosomal preparation. So what it indicates, every organism have got a default body structure. So in the case of human, the default body structure is feminine. It is the pattern of a female. So many organisms, the hormones or the genes are resisting to become another one. So this is how the evolution has put forward individual organisms. We will see them when we study about the dosage compensation, etc. So coming back to alternative splicing, the potential for an increase in the phenotypic diversity without increasing the overall number of genes. This is the foremost advantage, that with less you can do more, because each protein is a functional molecule. It is achieved by altering the pattern of exons that are spliced together. So which are the exons that should be spliced together or joined together depends on which tissue or which organism you are referring to and which gene you are referring to. Different proteins can arise from the processed mRNA from a single gene, because each RNA have got different coding sequence and each of them can perform different tasks. So alternative splicing can occur either at specific developmental stages or in different cell type. Sometimes a given protein is needed only for maybe for 2 days during the development. Then dedicating a full-fledged gene for that can be, counterproductive. So instead, make an alternative spliced form of that particular protein, make use of it, and rest of the time do not go for that alternative spliced form, because that gene can perform a different task throughout the organism's life. So this is how you make use of alternative splicing during the development. Of course, we cannot make use of at our will. It is how nature has evolved, or an organism have evolved during the evolutionary time scale. Let us see an example. The calcitonin gene yields an mRNA that synthesizes calcitonin, which is in the thyroid gland. Thyroid gland is somewhere here near your neck, and we know thyroxine is an important hormone which is important for the, if you don't have thyroxine during your childhood, you end up, I think that disease is called cretinism, you will be dwarf and you will not have proper intellectual development. But if you have dwarfism because of growth hormone deficiency, then you will be short but you will be normal. You are intellectually not challenged. But thyroxine is so important. And you may have heard many people will be taking thyroid tablet, that T3 TSH, you would have said that metabolism will be very low. We call them as hypothyroid condition, means their body has proper thyroxine production but not adequate. So you have to supplement with tablet. It's very important. Many times for miscarriage during pregnancy, etc., will be a major problem if the thyroid hormone levels are under the normal level or below the normal level. So it's very important throughout the lifespan for maintaining the body's metabolism. So calcitonin is a gene that is important in regulating the calcium balance in the body. So it is normally expressed in the thyroid gland, whereas the calcitonin gene related peptide, it is called CGRP, which is expressed in the brain. Both are coming from the one gene. And two proteins with distinctly different function in different tissue. So one is in the brain, another is in the thyroid. So coming from the same gene. And the alpha tropomyosin mRNA have at least 8 different alternatively spliced alpha tropomyosin mRNAs. This is something interesting and very important for us to keep in mind, because several structural proteins, because tropomyosin, troponin, these are all structural proteins in the muscle. Like you have muscle contraction, you are able to move your hand, finger, your facial muscles are moving. I am able to give a lecture. All credit goes to muscle contraction happening at different parts of my body. So you need to have structural proteins to perform the task. So heart have a different type of muscle, and my skeletal muscle have got a different type of muscle, my blood vessels have a different type of muscle. So we will not go into those details, but understand structural proteins often need more diversity than an enzymatic protein or a hormonal protein, because they have to perform differentially in different tissue. So one example is alpha tropomyosin, which is a structural protein. Structural protein means which provides structural feature or that provides function because of its structure. If an enzyme, say salivary amylase, it doesn't provide any structural advantage, it provides a functional advantage. When you eat chapati and if you chew it, salivary amylase act onto the starch and convert it into glucose. It's a function. Whereas structural protein, it has to be there in order to perform a task. So structure related function. So we usually call them as a structural protein. So alpha tropomyosin is a structural protein, but it can give rise to, mRNA can give rise to 8 different alternatively spliced alpha tropomyosin mRNAs and subsequently that much different proteins. Let us see how this is done. We will see the calcitonin example also. So this is an example of rat calcitonin. It has got a 5' non-coding exon and it has got common region coding exon C1 and C2, and then you have another big exon and followed by a poly-A tail, and we call it calcitonin coding exon. And then you have downstream, remember you have a poly-A site, and then downstream you have a unique exon, that is CGRP coding exon, and then you have a CGRP non-coding exon, you have a poly-A site. So what do we understand? You have a common region, first exon, second, third, fourth, like that, but you have 2 different poly-A sites. So during transcription, what it happens, the pre-mRNA, you have this 5' non-coding exon capping happened, and you have proper splicing C1 to C2 and calcitonin specific, and the poly-A site is made use of, and it goes smooth, and you end up getting calcitonin mRNA. In the thyroid cells, you have 5' cap C1, C2 calcitonin and poly-A tail, it goes smooth. Whereas in brain, what happens, it completely skips this calcitonin, it includes as a big intron. So it rather makes use of the CGRP. Remember, it has got this non-coding exon C1 and C2 common, non-coding exon C1, C2 common, but this calcitonin region is now replaced by CGRP because calcitonin region is skipped, and this polyasite was not used in the brain because it is masked, and the second polyasite is made use of, and you end up getting CGRP RNA. Then comes alternative splicing and its other features. Many defects in the beta globin genes are known to exist, leading to beta thalassemias. You may have heard about this disease condition because pregnant women are often screened for the embryonic baby whether it is vulnerable for beta thalassemia. So parents usually their blood sample will be tested, so to check it whether they are vulnerable for beta thalassemia by gene sequencing, etc. Some of these defects can be caused by mutations in the sequences of mRNA required for intron recognition, and therefore it can result in abnormal processing of beta globin primary transcript. When beta globin is abnormally processed, you will have a defective, less functional or non-functional hemoglobin. So beta thalassemia arises from this defective hemoglobin structure. Like I gave an example of sickle cell anemia, and like that beta thalassemia is also a structural defect in the globin. So globin is an important component of hemoglobin and also in the formation of the RBCs. And let us see some real classic example of Drosophila, and the gene name is DSCAM. What is DSCAM? Drosophila cell adhesion molecule. That is what DSCAM gene. It contains thousands of possible splice variants. So genomic DNA and the pre-mRNA, if you are seeing, you have got exon 4, exon 6, exon 9 and exon 17. They participate in a diverse variety of alternative splicing. That means exon 4 can contribute 12 alternatives, exon 6 can cause 48 alternatives, exon 9 can bring in 33 alternatives, and exon 17 can bring 2 alternatives. In the end of the day, once the mRNA is formed, it can bring up to 38,016 alternative spliced product, which is larger than human genome in itself. Human genome is supposed to have 23,000. So one DSCAM gene in Drosophila is capable of encoding up to 38,016 variety. So remember, it has got plenty of exons, all are not participating. 4 exons leave a total number of possibilities. What are they? Exon 4, exon 6, exon 9, exon 17. With just 4 exons contributing to alternative splicing itself, you can bring in around 38,000 plus varieties. The protein variants are important for the wiring the nervous system and immune response. So during the development of a complex organism like human, you can imagine how much diverse, how much variety of proteins will be needed if a nervous system of Drosophila require the cell adhesion molecule DSCAM of up to around 38,000 different varieties. So it is just a question of being detected. It is not a question of if. No question exists if there is an alternative splice method of introducing complexity in human. It is just a question of how many and when these are alternatively spliced. So let us see the splicing once again for our easier understanding. During the mRNA splicing, the first step is the 2' hydroxyl of the adenosine residue at the branch point attacks the phosphate bond at the 5' splice site to release the 5' exon and to form an intermediate lariat of the intron. Intron is still remaining attached to the second exon, which at the 5' end of the intron is attached to the adenosine at the branch point, that is in the intron. And this pairing is 5' 2' phosphodiester bond, that is the lariat. And during the second step, the 3' hydroxyl of the 5' exon, because 5' exon is now completely free, but it is not releasing, credit goes to the snRNA or the spliceosomal machinery. Exon attacks the phosphate at the 3' splice site, resulting in the ligation of the 5' and 3' exon. Here comes the catch. Now the 3' OH of the 5' exon, where is it going to start its hybridization or its fusion or its ligation or its splicing? Where is it going to start? And wherever it is starting, it will include the portion as the intron. So first step is the lariat is formed because of the adenosine at the branch point, and now the first exon or the 5' exon decides where it should fuse. So cell can have different ways of regulating the second transesterification reaction or the second ligation reaction. Accordingly, you can decide an exon should be longer, shorter, or where the second fusion should happen. That is why we are revisiting the splicing once again, although we have already addressed the splicing. So now if you see closely, this spliceosome is a large particle, which we know. It is not a simpler thing, and it is a highly it's a highly organized structure, and it is regulated in a stringent manner, and it may sound like everything is perfect, and to some extent it is perfect, everything is perfect, but splicing defects can also happen, sometimes with a huge price, and we will see some example. Let us see more in detail that why we call spliceosome as complex. Some examples will make you understand better. Around 2.1 megadalton mass is there, and around 30 different proteins are present, which is constituting 17% of the mass, and this is done by 5 snRNA, which is around 3.3 megadalton and 27% of the mass. Then again you have 41 proteins, and which is constituting to the snRNP, which will add to around 2.2 megadalton and 18% of the mass, and 70 splicing factors which is around 4.7 megadalton and 38% of the mass. So we should understand spliceosomes are forming only a minority compared to a large number of proteins that can bring in variations during the splicing process. So we are seeing here quickly the splicing, that is exon 1, exon 2 and the intron 1, and you know U1 and U2 binds. U1 binds in exon 1 and intron 1 boundary, U2 binds in the branch point, and then the complete spliceosome assembled once U1 and U2 are paired and cleaves the transcript at the 5' splice site, because the assembly of the spliceosome causes a bending, because unless the intron is bent, you will not be able to form the lariat, and the lariat is formed here. The 5' end of the intron 1 is joined to the A in the branch point and to form the lariat structure, and the U1 and U4 are now released, and now you have to have the second transesterification reaction. The 3' splice site is cleaved at the 5' end of the exon 2 and is joined to the 3' end of the exon 1, and the lariat shaped intron is now released and it is free to go, and the exon 1, the 3' OH of the exon 1 has to be very crucial in deciding that where the fusion has to happen at what point onto the exon. So the nuclear splicing involves the so-called transesterification reaction. You are seeing this splicing in a less complex structure. You have got this A, it has got this branch point OH, and this OH is 2' OH. 2' OH participate in the first transesterification reaction, and then it goes into the second transesterification reaction here, and you end up getting fusion of these two exons, and the lariat is released, and the branch site structure is shown here, but the red color label to the A is the branch site A, and the GTAG rule is compulsory. In RNA it is GUAG rule, and let us see this picture shows how important they are. This blue color one is exon, and this little dull pink color portion is the intron, and this exon is having ending A and G, exon, okay, and 64% is A and 73% is G. Now let us see the G and T in the intron. 100% is G, 100% is T. That means that rule is not flexible. And same way you see the other end of the intron. 100% is A in the towards the end of the intron, and 100% is G. That rule is not fixed. On the other hand, you see here this is an N. First base of an exon is an N, means it can be any nucleotide. There is no compulsory. That means this exon are free to fuse as and when it required, wherever it required. There is no rule that it can have variation. So this adds to the complexity. So GTAG rule describes the presence of this constant dinucleotide at the first and the last two positions, GT and AG positions of the introns. And the splice sites are the sequences immediately surrounding the exon intron boundary. 5' splice site, 3' splice site. The splicing junctions are recognized only in the correct pairwise combinations. So we will revisit more about the splicing in the subsequent classes. Thank you.